Hello everyone, myself Ajit Kumar. I am doing first year MSc Biotechnology in VIT University. I am going to present a presentation on Bacillus Anthracis, which is a rare and potentially fatal disease, which is also known as bioterrorism threat. The content in the presentation are introduction, history of Bacillus anthracis, morphological and structure, pathogenesis, diagnosis and treatment, prevention and control, or we are going to see now. Bacillus anthracis is a bacteria that has played a significant role in the history of microbiology from its discovery in the late 19th century. It also uses as a biological weapon. The microbe has been captured the attention of scientists, policymakers, and the public alike. In this presentation, we will explore the fascinating world of Bacillus anthracis from its morphology and structure to its pathogenesis and treatment. We also discuss the importance and prevention and control measures to combat this deadly bacteria. History of Bacillus anthracis. The first recorded report of anthrax was during the 5th Egyptian plague in 1500 BC, responsible for the death of a large amount of livestock. The 6th Egyptian plague, also known as plague of cutaneous anthrax, was responsible for many deaths. The Black Bane in Europe during 600s killed well over 60,000 cattle and typified anthrax symptoms. In 1863, French biologist Casimir Joseph Devine makes Bacillus anthracis the first microorganism to be conclusively linked to the disease. In 1876, Bacillus anthracis, isolated in a pure culture for the first time by Robert Koch, was also called wool sorter disease, Bradford disease, and rag picker disease. Now we are going to see about morphology and structure of bacteria. Bacillus anthracis is a rod-shaped bacterium that typically appears in chain under the microscope. It has a thick protective outer layer called a capsule that helps it evade the host immune system. The bacterium also produces spores that can survive in harsh environment for extended period of time. Under high magnification, the surface of the Bacillus anthracis appears rough due to the presence of hair-like structure called villi. These structures play a crucial role in the bacterium's ability to attach the host cell and causes infection. Additionally, Bacillus anthracis possesses a complex cell wall made up of multi-layer which provide further protection against the host immune system. There are two types of antigens that can cause Bacillus anthracis disease. One is antigen called important antigen, which is also known as capsular antigen, and one more antigen or known as another antigen, which is also known as cell wall polysaccharide antigens or somatic protein antigens. Anthracis remains susceptible to many antibiotics including doxycycline, and it also resistance to penicillin G and amoxicillin was present in 11.5% of the isolates, suggesting that these antibiotics should not be used in a prophylaxis or in the treatment of humans without susceptible testing. The bacteria spores can be viable for over years in a soil. Autoclaving can kill the anthrax spores. Potassium permanganate can kill anthrax spores, and it is more susceptible to many antibiotics like penicillin. Now let us see about pathogenesis of B. anthracis. Bacillus anthracis is a highly virulent bacterium that is responsible for causing anthrax, a potentially fatal disease in humans and animals. This pathogenesis of Bacillus anthracis involves several key steps including spore germination, bacterial replication and toxin production. Once the spore of Bacillus anthracis are inhaled or ingested, they can quickly germinate and begin to replicate within the host body. The bacteria then produces a variety of toxins that can cause tissue damage, inflammation, and other harmful effects. In severe cases, these toxins can lead to organ failure and death. In animals, it can be infected rarely by contact with another animals. Ingestion of spores very common in animals. Infected animal discharge more anthrax bacilli from mouth, nose and rectum region. Sporulation in a soil can survive for years sources for other animals. This bacteria can also infect human beings and it is also known as human anthrax. It can be get infected by accidental inflammation, secondarily infected from diseased animal. There are three types of clinical disease which can infect humans. One is cutaneous anthrax, more common in human beings. Secondly, pulmonary anthrax, which is very less common. And the third one is intestinal anthrax, which is rare and it can be found in human beings, where it can also affect the intestinal region. First, we are going to see about cutaneous anthrax. The cutaneous anthrax is disease which can be caused in a skin of a human being, which is bacteria infect your body through a wound in your skin. Cutaneous anthracis is the most common and least deadly form. Veterinarians and people who handle animal wool, hide and hair are the highest risk, which can germinate and multiply in an infected site. Spore enters through the abraded skin. Localized lesion can be found on a skin, face, neck and hand region. In pulmonary anthracis, the bacteria can affect the lungs in a human being. In this type of disease, where people who breathe in anthrax spore can develop this deadly form of anthrax, it can also cause severe breathing problems and death inhalation. Anthrax is sometimes called wool sorter disease because people who work in a wool mill as well as slaughterhouses and tannery workers may inhale spores from the infected animal who can easily get affected by this pulmonary anthracis. The intestinal anthrax can affect the intestinal region of human beings. This type affects people who eat undercooked or raw meat from an infected animals. The bacteria affect your esophagus, throat, stomach and intestines. Gastrointestinal anthrax is rare in United States. Based on producer, vaccinate livestock against anthrax and identify sick animals before slaughters. To detect Bacillus anthracis, there are several ways and laboratory techniques which can detect Bacillus anthracis. The first one is cutaneous anthracis can be detected by pus from pustules. The pulmonary anthrax can be detected by sputum test. The intestinal anthracis can be detected by stool or urine test, and the septicemia anthracis can be detected by blood test. Microscopic test to detect Bacillus anthracis. One is gram stain for bacilli demonstration, McFadden's reaction for capsule demonstration, and spore staining for spore demonstration. The culture media used in a laboratory to detect the Bacillus anthracis and the medias are selective media, pelt media, polymyxin, lysozyme, EDTA, and thallose acetate. Gelatin liquefaction is a test that are used to detect Bacillus anthracis by using test tubes and medias, where maximum liquefaction on the surface then at the bottom, which indicates the presence of Bacillus anthracis, and also inverted R3 appearance. The another laboratory test that used to detect Bacillus anthracis is a nitrate detection test, which indicates media. When a media turns into a transparency color or white color, which is negative nitrate, or known as absence of Bacillus anthracis. In this case, where if the media turns into red color, which is positive, where presence of anthracis can be confirmed. Diagnosis of Bacillus anthracis. The diagnosis of Bacillus anthracis infection can be challenging, as the symptoms are similar to those of other diseases. However, laboratory tests such as blood cultures and PCR can confirm the presence of the bacteria in the patient's body. The diagnosis can also made by the doctor will ask you what kind of work you do along with other questions to determine the likelihood of your having been exposed to anthrax. He or she will first want to rule out other more common conditions that may be causing your signs and symptoms such as flu and pneumonia. The standard treatment for Bacillus anthracis is the antibiotics such as ciprofloxacin or doxycycline, which are single antibiotic or combination of antibiotic, and the length of treatment will be most effective for you depends on how you were infected with anthrax, your age, your overall health, and other factors. Treatment is more effective when started as soon as possible. Since the 2011 attacks in the United States, researchers has developed antitoxin therapies for inhalation anthrax. Instead of going after the bacteria that causes the disease, this medication helps to eliminate the toxin caused by the infection. Anthrax immunoglobulin also may be used to neutralize the toxins. These medications are given in addition to antibiotics and are available to doctors through the US Centers for Disease Control and Prevention. In some cases of injection, anthrax have been successfully treated with surgical removal of infected tissues. Although some cases of anthrax respond to antibiotics, advanced inhalation anthrax may not. By the later stages of the disease, the bacteria have often produced more toxin than drug can eliminate. Along with antibiotics, people with anthrax may be treated with intensive support, including ventilators, fluid, and medicine to tighten blood vessels and raise blood pressure. Prevention and control for Bacillus anthracis disease. Preventing and controlling Bacillus anthracis infection is crucial in maintaining public health. One of the most effective methods is through vaccination. The anthrax vaccine is highly effective in preventing infection and is recommended for individuals who work with animals or in high-risk environments. Additionally, proper hygiene practice such as washing hands and cleaning surface can also help to prevent spread of the bacteria. In the event of an outbreak, antibiotics are a primary treatment method. It is important to seek medical attention immediately if suspect you have been exposed to Bacillus anthracis. Early treatment can greatly improve your chances of recovery and prevent the spread of the disease.